Confirm you can hear me? Let me check my speaker. All right, before we begin, are there any questions? I'm not hearing anything, just to let you know. See if I can. My computer is moving a little slow because it's still working on the lecture. All right, let's begin with the lab. I'm just going to assume everything is well. You can text me if you wish. Let me share my screen here. Now, if you text me, I won't be able to see it, but so I'm just going to go over the material from chapters 1 to 8, what the exam might be going over. And this material you've been quizzed on already, so it'll be a little bit of a review, so I'm going to be very quick about it. Chapter 8 has a few more questions than most of the other chapters, but chapter 8 is a big chapter covering microbial genetics. You should always know the terms. You should be able to describe DNA replication, RNA transcription, and protein translation. And you should understand the different types of mutation. All right, that's just terms. You should know the terms. You should know the central dogma in biology. That's that information starts in DNA and then flows to RNA in transcription, and then that information moves into or flows in translation to protein. So the DNA contains information and it's coded in the genomes. The RNA contains information, it's contained in the nucleotides of the RNA, and then that's translated into the protein. And that information is in the sequence of the amino acids in the protein. And that's the flow of information within a cell. You can have a flow of information between generations of cells, or if you're talking about humans, between generations. And that is the information in the parent cell flows, well, it replicates and then flows to two daughter cells, where each daughter cell gets the same information as the parent DNA. Now, in humans, you got two adults, the mother and the father, which then would replicate and divide into half, and then half in the egg and half in the sperm, join to make the zygote, which then would grow up to become the baby. And that's the flow of information between generations or generations of cells. The flow of information between cells of the same generation is DNA leaves one cell, the donor cell, and then usually it's outside in the environment, naked DNA, which comes to the recombinant cell, which takes up that DNA and then becomes recombined with DNA of this donor cell, as well as most of the DNA from the original cell. So that's DNA between cells of the same generation. You should know about DNA replication. I'm not going to go through the video, but you should just know that DNA always is made 5' to 3'. And so one of the strands is made continuously, the leading strand. The other strand, the DNA is made in short fragments going the opposite way the strand is going. And these are called Okazaki fragments, so that the DNA is replicated in short strand fragments. And you need DNA polymerase continually binding to the lagging strand, as well as DNA ligase ligating the fragments together. The three important enzymes in DNA replication are helicase, opening up the double-strand DNA, making it single strand. The DNA polymerase, taking the single-strand DNA, using as a template to put down the new DNA on the new strand. And then the third important protein is the ligase, and that's linking these strands of DNA together, which on the lagging strand, there's a lot of strands that the ligase has to link together. On the leading strand, if the DNA polymerase never falls off the DNA and starts at the end of the chromosome and continues transcribing to the other end of the chromosome, there will never be use of the ligase. However, oftentimes the DNA polymerase falls off, another DNA polymerase will bind and then continue the process, and then the ligase has to link the two fragments together, meaning the original fragment and then the newly added fragment. The original fragment was put down by a DNA polymerase that fell off. You should know about transcription. That's the making of RNA from DNA. Only the genes are transcribed when we undergo transcription. None of the non-coding DNA is transcribed. In DNA replication, all of the DNA is replicated. DNA transcription begins when the RNA polymerase binds to the promoter, a region of DNA upstream of the gene, and the promoter codes for the DNA polymerase to bind to the DNA at that site. And then the promoter moves down the DNA, transcribing RNA until the RNA polymerase reaches the terminator, a region at the end of the gene. The terminator codes for the RNA polymerase to fall off the DNA, and then that'll stop RNA transcription. You should know about protein translation, and that the messenger RNA encodes the information, and the messenger RNA is read in groups of 3 nucleotides that we call a codon. The first start of the messenger RNA is ignored. The ribosome will move down the messenger RNA, and it will not begin protein translation until the ribosome receives a start codon, AUG, and the start codon tells the ribosome to start protein translation at this site. And at this site, AUG also codes for the addition of methionine, that amino acid. So AUG is doing 2 things, telling the ribosome to begin protein translation here, and then also telling the ribosome to add the amino acid methionine. And then the ribosome will move down the messenger RNA, reading each codon, adding the appropriate amino acid until the ribosome reaches one of the 3 stop codons. The stop codon will then tell the ribosome to disassemble, and that will release the messenger RNA as well as release the newly synthesized protein. Now, I've stated that the ribosome is reading the messenger RNA, and the messenger RNA is read in groups of 3 nucleotides that we call a codon. Most of the amino acids have more than one codon coding for that amino acid. There are 2 exceptions, methionine and tryptophan. There's only one codon coding for those amino acids, but all the others have at least 2. Some of them have 4. Oops, sorry, that's one that has serines, one that has 4. Leucine has 6. So some have 2, some have 4, and some have 6. There's only 2 that have only one codon coding for that one amino acid. Any questions about any of that? Oh, I did mention that the ribosome is reading the messenger RNA. The ribosome is actually not reading the messenger RNA. It is moving down the messenger RNA. What's doing the reading of the messenger RNA is the tRNA. The tRNA comes in the hole in the ribosome, which then stabilizes the tRNA, and then the tRNA, the correct one, will bind to the codon because it has an anticodon which actually hydrogen binds to the codon, and the ribosome stabilizes it so that this is stable because the at most that would be 9 hydrogen bonds binding. That's not very much, and it's not a very strong link, so the ribosome stabilizes this link. But actually, it's the tRNA that's really doing the reading of the messenger RNA. All right, you should know about bacterial gene expression. It's regulated by the operon. An operon is like a super gene, including the gene of interest, which is a structural gene, and then the controlling regions around that structural gene. So all operons have at least one structural gene, one operator, and one promoter. And not shown here, they also have a terminator. The promoter we already talked about is the region where the RNA polymerase binds to the DNA and then moves down the DNA to transcribe the DNA into RNA. The operator is a regulatory region controlling the expression of the structural gene. What the operator does is it's the binding place for a regulatory protein. It's made at a different gene. The regulatory protein can bind to the operator and then shut down the expression of this structural gene. Well, how that happens is when the structural gene, the regulatory gene is bound to the operator, the RNA polymerase cannot move further. It's stuck at the operator, positioned by the regulatory protein. That means the structural gene will not be transcribed. The structural gene is the gene that we normally talk about when we're talking about a gene. It is, in the case of the lac operon, the Z gene codes for the enzyme lactase. All eukaryote operons only have one structural gene. Bacterial operons may have one structural gene, or they may have a couple of structural genes. If they have a couple of structural genes, all of those structural genes will be related. Like in the case of the lac operon, all of these structural genes are related in the metabolism of the sugar lactose. The Z gene encodes for digesting the sugar lactose by the enzyme lactase. The Y gene codes for the transport protein that allows the sugar lactose to be transported into the cell. The A gene helps code for converting the sugar beta galactose to the sugar glucose. If you don't know, lactose is the sugar beta galactose hooked to the sugar glucose. And to metabolize the lactose, you have to convert it to the sugar glucose. As glucose is the base sugar. Any questions about the lac operon? All right, I've already explained how the operator works. The regulatory gene, which is another operator up here, makes a repressor protein which binds to the operator, stopping transcription of the Z and the Y and the A gene. You want a way to turn on the operon or the lac lactose genes, like lactase. And how you do that, there is an inducer which binds to the repressor protein. In the case of the lac operon, the inducer is the sugar allolactose. Allolactose is an isomer of the sugar lactose. So whenever you have the sugar lactose around, you have allolactose around. And allolactose will bind to the repressor protein, inactivating it. And if there's no repressor protein, then it can't bind to the operator. And then the lac operon is turned on because the polymerase will bind to the promoter and then transcribe the Z, the Y, and the A gene. Right here, just to mention the regulatory gene, when it's bound to the operator, its binding is stoichiomic, and so every once in a while it falls off. If allolactose is around, it'll then bind to the repressor protein, inactivating it, and then it can't rebind to the operator, and then all the other repressor protein is inactivated by the allolactose. And so that's how the lac operon turns on. Meaning I'm trying to tell you that the regulatory protein, when it's bound to the operator, it has to fall off on its own because the inducer cannot bind to the regulatory protein when it's bound to the operator. All right, any questions about the lac operon? You should know about the different types of mutations. There are two ways a mutation can occur. A mutation is just a change in the genetic information. You can have a mutagen. This is an agent that actively causes a mutation. And you can have spontaneous mutation that occurs in the absence of a mutagen. Just when the DNA is replicating itself, a mistake can be made, and that would be a spontaneous mutation. There are different types of mutation. There's a base substitution where one codon is changed in the DNA, one nucleotide is changed, and it can give rise to a different type of mutation. But the point is the DNA nucleotide is changed, and so that's a base substitution. A frame shift mutation is where one or a multiple of bases is either added or subtracted, and that changes the DNA from that point on, because the codon, if you add a nucleotide, the codon reading frame will be totally changed from that point. And that's why it's called the frame shift mutation. What is not a frame shift mutation is if 3 bases are added or 3 bases are removed, or some multiple of 3, because that will only change that location, which would only change one amino acid. It does not change the entire reading frame of the DNA. Any question about any of that? And then the third type of mutation is a macromutation, where an entire region of a chromosome is either added or deleted. A macromutation is not in your textbook. A Down syndrome is a classic example of a macromutation, where there's an entire extra chromosome in an individual born with Down's syndrome. A base substitution is where you change one nucleotide and it changes the amino acid in the protein. A silent or a neutral mutation is where you change the DNA nucleotide, but you do not change the amino acid in the protein, such as when UUU, coding for phenylalanine, is mutated to UUC. It changes the DNA, but it does not change the amino acid in the protein. So there's no change or net effect in the protein. And that's why it's called a silent or a neutral mutation. And then there's a missense mutation where you change the nucleotide, but instead of it changing the amino acid to a different amino acid, it changes the amino acid to the stop codon. And then you'll get a truncated protein. So instead of getting methine, lysine, phenylalanine, and glycine, oops, excuse me, that's a missense mutation. Let's go on to the base. Nonsense mutation is where you change the amino acid and get a truncated protein. So instead of having methionine, lysine, phenylalanine, glycine in the stop codon, you change the AAG to UAG. You don't need to know that, by the way. Coding for the stop codon, and then you get a truncated protein. Usually that inactivates the protein, by the way, but it does depend on where the stop codon is added. What did I talk about? Oh, base substitution. Yeah, when I'm talking about a base substitution changing the amino acid, that's called a missense mutation. All right, talked about a frame shift mutation. There's a positive direct selection, and that's where mutant cells either grow or appear different, so we can easily recognize them as different. A negative selection happens because the mutant cells do not grow. Here's a case of a negative selection where we have a bunch of colonies, stamp it onto two different media. On the plate containing the amino acid histidine, we get the cells growing. On the plate lacking the amino acid histidine, there is no growth. And so this is negative selection to detect a mutant. These cells here are mutated. They'll only grow when you supply the amino acid histidine. You should know about recombination, the exchange of genes between two different DNA molecules, such as when crossing over occurs. Recombination is important because it can bring two genes together that were not together previously. Once the mutation creates a new genetic variation in the population, the new genetic variation can occur in other cells by obviously mutation, but it can also occur by vertical gene transfer or horizontal gene transfer. Three different types of horizontal gene transfer: transformation, conjugation, and transduction, where pre-existing DNA in the population is spread to another individual by some means, transformation, conjugation, or transduction, spreading around the pre-existing information. The reason why this is important is horizontal gene transfer, all three of these, happen 10 times more often than spontaneous mutation. Transformation is where DNA comes out of one cell, is naked DNA, is combined or joining with the recombinant cell, and then that recombinant cell expresses some or all of that DNA that came out of the donor cell. And this is important in the sense that we're giving this cell, which has never had it, a little case A gene. And like little case A could code for antibiotic resistance, and then that would make this cell now antibiotic resistant. Conjugation is where a plasmid moves from an F plus cell into an F minus cell. The plasmid itself codes for the production of a sex pilus on a gram negative cell. Conjugation can also happen by a mating bridge, in a gram-positive cell. What happens with sexual conjugation in a gram-negative cell with a sex pilus is the sex pilus goes out and skewers an F minus cell. It can bring the cells together, but the cells don't need to be this close because that sex pilus is hollow, that the plasmid can leave the F plus cell and then move to the F minus cell. If you notice, the plasmid codes for the production of the sex pilus. And then when this happens, that F plasmid replicates and then sends a copy of the plasmid through the sex pilus into the F minus cell. That F minus cell now has the F plasmid in it. The F plasmid will cause the F minus cell to grow a sex pilus, and then that F minus cell will become F plus cell, meaning it'll go out and skewer an F minus cell and cause sexual conjugation to happen. Any question about conjugation? Transduction occurs with a virus spreading around pre-existing genetic information. When a virus infects a cell, it will inject its genome into the cell and then will degrade the host chromosome. Let me blow this up. The virus will then replicate in the cell, make the protein coat, assemble the virus, but occasionally the virus makes a mistake, and instead of adding viral genome, viral DNA, let's say, to the virus here, it will add the host chromosome DNA to the virus. Now, when this cell infects this virus, goes on and binds to the cell and infects it with the DNA, that's not viral genome, that's the genome of this cell. That genome can then recombine with the host chromosome, bringing the DNA of this portion of this cell into this cell, giving this cell new genetic variants, such as if this cell were to get on this DNA coming from this cell, a antibiotic resistance. Any question about any of that? Okay, you should know that plasmids are small particles of DNA holding only a few genes, different from the chromosome, which holds hundreds of thousands of genes, at least thousands of genes. Plasmids are important because they're more likely to be shared between two cells than the chromosomal DNA. You should also know about transposons. These are jumping genes, regions of DNA which move around in the host genome, meaning that in a eukaryote cell, the transposon can actually jump out of one chromosome and then re-enter another chromosome. In bacteria that only have one chromosome, it just moves from one part of the chromosome to another part of the chromosome. You should know the structure of a transposon shown here, and you should know a complex transposon usually has a transposable unit on each end of the complex transposon, and then a gene in between, such as TN5 carrying the kanamyosin resistance gene. This kanamyosin or transposon can move out of the chromosome, move into a plasmid, and then this plasmid would be more likely to be shared between bacteria cells in a population, moving around the kanamyosin resistance gene. Any question about any of that? All right, I didn't mean to be that long on chapter 8, but it is an important chapter. I guess I'm going to leave
I
that.
have a question.
Uh-huh. What's your question?
It's about the final. If you know,
Uh-huh.
are you able to open it up on the 17th, the morning, because the graduation for Clark is on the 18th.
This is a little late requesting for a time change on the final. I would have to notify everyone and get their approval, so I'm not likely to do that. I can put it in the an announcement, but unless everybody replies with a yes, I'm not going to move the time. Okay? I'm sorry about that. Like I said, you're a little late on asking for the change. I didn't quite understand when the graduation is and when the final is, but you have 24 hours in order to take the final. So you should be able to take it even if you go to the graduation ceremony.
Okay.
Okay. All right, let's move on to chapter 6. And I promise I'll be shorter on this one. I'll be shorter on all the others except for chapter 1. Chapter 6, obviously know the terms, understand the various requirements of cells for growth, understand the 4 phases of growth, and then compare the different methods for measuring microbial growth. Here are the different methods for measuring microbial growth. The direct methods are the plate count, the filtration method, the most probable number, and the direct microscopic count. I won't ask any questions about the most probable number because I've never done the most probable number. It also requires the use of mathematics. And then the indirect methods, turbidity, metabolic activity, and dry weight. Any questions about the methods for measuring microbial rate growth? If not, that's it for the chapter. Let's move on to chapter 5. Obviously know the terms, understand what feedback inhibition is, understand what aerobic respiration is, its steps, and how it differs from anaerobic respiration. One thing that's not on here is we also discuss fermentation. You should know what fermentation is. Any questions about chapter 5? Move on to chapter 4. You should know the functional anatomy of prokaryotic cells. I'm unlikely to ask you anything about eukaryote cells, with the exception of the endosymbiotic theory. Because we did discuss that in this class. So know the endosymbiotic theory. You don't really need to know anything about the function of eukaryotic cells. You should know how gram-positive and gram-negative cell walls differ, and then describe the importance of taxis in bacteria. You should know the different shape that bacteria cells take, 3 main shapes shown here. There are different shapes, but I'm not going to ask any question about the square shape or rectangular shape and the star shape. You should know that most bacteria have one shape, so they're monomorphic, but there are a few species that are pleomorphic, where the cells have many shapes. You should know the different types of spirals, spirochete, spirillum, a vibrio, and you can call that a rounded or curved rod as well. You should know the different cellular growth arrangements. Cocci have the most cellular growth arrangements. There's a singlet, there's a diplococci, there's streptococci, there's tetrads of cocci, there's sarcenia of cocci, and that's 8 cells, and then there's staphylococci. Staphylococci literally means cocci, looking like a shape of grapes. For bacilli, they have singlets, diplobacilli, streptobacilli, and then a palisade. Palisade is where they're linked together on their long end as opposed to on their short end. Any question about cellular growth arrangements? Here's a question for you. Which cellular growth arrangements do the spirochetes have? Can anyone answer that? First of all, they are monomorphic, spirochetes. Is there a diplospirochete? All right, there's no diplospirochetes. The spirochetes only have one shape, and that's the singlet. All right, any questions about chapter 4? Not, let's move on to chapter 3. Understand the terms and the goals of this chapter, or the objectives. Understand the sizes of organisms and the properties of microscope. Understand the major differences between a light microscope and an electron microscope. You should understand why staining improves resolution. Improves resolution only by increasing the contrast between the cell and the background that the cell is on, meaning the slide. There are 3 types of stains. You should know what a simple stain is, a differential stain, and a special stain. You should also understand the Gram stain, its procedure, and the basis of its differential staining. I guess you should understand why we use the metric system. It's because science and medicine use the metric system. And this map is showing you all the countries in the world that are not currently using the metric system. And 2 of them are actually slowly converting to using the metric system. I think it's these 2, but I'm not sure which 2. The United States is one of them. All right, any questions about chapter 3? All right. In chapter 2, there are no direct questions regarding chapter 2 on the final. You do have to understand the terms of chapter 2 and the concept of chapter 2. If I give you a figure and it has a chemical on it, you have to understand that. But I'm not specifically asking anything about chapter 2. On chapter 1, we introduced a lot of introductory concepts in chapter 1. Such as Koch's postulates, pasteurization. We introduced the cell wall briefly in this chapter 2. We also introduced the endospore in chapter 1. So all of these topics were first introduced in chapter 1, and then we went on and talked about them, most of them about 3 times further, like Koch's postulate and endospores. We talked about 3 times further. You should also understand the brief history of microbiology, where you know who found the major finds in microbiology and why that was important. Like Louis Pasteur came up with the first method for getting a pure culture, but it wasn't easy to accomplish. And then Dr. Koch came up with an easy way of getting a pure culture, and that's just streaking out your cells on a solid media to get isolated colonies. You then take from that isolated colony and then streak it out again to get isolated colonies. And the cells that have gone through 2 colony isolation events are greater than 95% chance of being a pure culture. All right, any question about that? I'm not going to go over the different concepts of history of microbiology, but let's go over a few things. You should know what bioremediation is. You should know that um, Carolus Linnaeus or Carl Linnae or Carolus Linnae, I think is how your textbook calls him, was somebody who started the scientific naming principle, also called the scientific nomenclature or the binomial nomenclature, where all species have two names, a genus name, human genus, and then the specific species epithet, the sapiens. To name the species, you have to get both the genus and the specific epithet name. This was so widely popular and so widely familiar that it's still in use today. He also started the classifying of organisms, but his classification system is no longer used. He classified all organisms into plants and animals, and as we know, microbes are neither a plant or an animal. You know about the domains. There are three domains. The domains are defined. Everything in a domain is similar to other organisms in their sequence of DNA. So the genetic data defines the domain. And if we ever discover a new animal or new creature, well, not new animal, new creature, like a new microbe, all they need to do is sequence the DNA, and then they'll determine which domain it belongs to. Domain bacteria, domain archaea, or domain eukarya. After the domains, we have the kingdom. There are different definitions for what a kingdom is. There's different numbers of kingdoms. We're just using the simple six kingdom approach where the domain bacteria is also the kingdom bacteria. The domain archaea is also the kingdom archaea. The domain eukarya has four different kingdoms: kingdom plantae, kingdom animalia, kingdom fungi, and then kingdom protista. Please understand kingdom protista is in the domain eukarya, but it's not a plant, an animal, or fungi. And so the kingdom protista is a collection of eukaryotes that are not plants, animals, or fungi. So the kingdom protista could be divided into different kingdoms if you so desired. We're not going to do that. We're just going to use the six kingdom approach. You should know that um, all cells have a cell type. It can be a cell type prokaryote or a cell type eukaryote. This is also called the superkingdom eukaryote or the superkingdom prokaryotes. And the superkingdom was in existence long before the domains were discovered. You should realize that viruses don't belong to a domain because they are acellular. All right, in the history of microbiology, showing here, this is the only date you need to know that the ancestors of bacteria can be found in fossils over 3.5 billion years ago. All the other dates are in here just for information. You should know that before the Mayans fermented a beverage, it was close to chocolate from cacao. The Babylonians wrote down recipes for making beer, and obviously before that, there had to be ancestors of the first life on Earth. You should know that Robert Hooke came up with the concept of cells. He looked at cork in the bark of the cork tree, which is called cork, by the way, and discovered cells. And that gave rise or began the rise of the cell theory that all living things are composed of cells. Yeah, he didn't propose that theory because when he looked at animals, he never found cells. You should realize that there were two hypotheses about how life originates: spontaneous generation, a vital force gets into organic matter, giving rise to life, and biogenesis, all living things arise from other living things. You should know about Francesco Redi, who did the first controlled scientific experiment testing whether life could spontaneously generate, and he found that there were no maggots in an open jar completely sealed or covered with a fine wire mesh, and there were maggots appearing in the meat in the open jar. So he did show the first controlled scientific experiment that at least maggots and any complex life higher than maggots could not spontaneously generate. However, people still believe that microbes could spontaneously generate. And we're not going to talk about why, but it's just because microbes were essentially everywhere. And whenever people made a media, it got contaminated by microbes. You should know about Antony van Leeuwenhoek. He's the first bacteriologist and the first I don't know, microbiologist. He looked at the first microbes. He built the best microscopes. He actually ground the lenses himself, and he was the best lens grinder. And he saw cells so small that were not seen again for 100 years, that the microscopes had to improve for 100 years before they could see bacteria, which you can recognize here. He found microbes wherever he looked. Louis Pasteur is the person who disproved spontaneous generation. What he did was he got a beaker, added media to it. The beaker had to have a long neck, and then he heated the glass there, bent it in a swan type neck, and then sterilized the media by boiling it. And then it's open to the outside air so the vital force could get in, and then found that the media was not having microbes growing in it, did not become turbid, unless he takes the media and puts it down here, because microbes could come down here by gravity, but they could not go up there, up this way, and then down that way by gravity. That disproved the theory of spontaneous generation. All right, this goes on. A few other people are important. I'm going to stop here and ask, are there any different questions? You do need to know some of the other concepts of the history of microbiology. I'm just stopping here to conserve on time. All right. Any question about chapter 1? If not, that's it. Good luck on the exam. And if you have any questions, ask. Remember, the Unknown project closes on Friday. That's the last time you can request a test. The Unknown project is due Saturday. All right. Bye.